think that's us. Hi, everybody. Welcome back, or welcome for the first time. This is the second part of our three-part webinar series looking at rapid-acting antidepressants from ketamine to psychedelics. So this is being run in partnership with the British Pharmacological Society and Transformation, a preclinical CRO. So as I mentioned, this is the second part of a three-part series. So this one focusing particularly on psychedelics in preclinical research and translatability. The format for anybody who wasn't here for the first webinar is quite informal, so I will pose discussion points or questions to the panelists and they'll answer in turn, and hopefully we can get a discussion going. And also just to mention that if you could please submit any questions you have into the question box, that would be great. Towards the end of the webinar, we'll try and address as many of those as we can. Hopefully we'll get plenty. First of all, I'm your chair. My name is Fionn Doherty. I'm a senior scientist with Transformation. We're joined today by three excellent panelists. Our first panelist here, Dr. Guy Higgins, who is the Chief Scientific Officer for Transformation and for Intervivo. He comes to us with a huge amount of experience. I suppose, if he doesn't mind me saying, decades of experience in the drug discovery space. Worked with several different pharma companies over the years, so looking forward to hearing from him. Our second panelist is Dr. Brigitte Robertson, who's the CMO with Delix Therapeutics, obviously a huge player in the psychedelics field. And again, somebody with a huge amount of previous experience in the drug development and CNS drug development industry, working with things like GSK and Takeda over the years. And then finally, our third panelist is Dr. John Huxter, who is the scientific liaison and the cognition lead at Transformation. And again, another person with a huge amount of experience in this space, an electrophysiologist by training, but now works, of course, across all kind of preclinical work for Transformation. Again, with a huge amount of experience in previous pharma, like Eli Lilly. So yeah, just want to thank all three of you for your time today, your valuable time. So just to move on to I suppose the first question or discussion point that I wanted to raise. And it's probably a broad kind of topic to start on, and maybe too broad, but just in general, thinking about preclinical drug discovery as a tool, maybe to think of it that way. What do you feel is kind of like the most important thing to get right with preclinical research, or where do you think what's its strongest strength or its strongest utility as a tool in drug discovery? Maybe Dr. Huxter, if you'd like to start off on that.
Oh boy, put me on the spot.
Yeah, if you can just wrap that up in a very succinct way.
I mean, the closer you can come to the model that you're trying to replicate, the system or the disease state, obviously the better. If you know the target, then I mean, knowing that your model expresses that target can go a long way to make sure you have translational success, I think. I mean, not all drug discovery has to be done in disease models. We need to be aware of that. There is no, true preclinical model of a human disease, certainly not in vitro, but even in vivo, At best, what we normally talk about is our ability to, to replicate symptomology. And that might be the appropriate, strategy going forward. And certainly, a lot of pharmaceutical companies are starting to take that approach. We recognize that a lot of the diseases that we're trying to treat are not, they're not unary. They're not a single thing even. So you take a patient with Alzheimer's or patients with schizophrenia. They don't all present the same way. They don't all have the same symptoms. Sometimes the pathology is not in the same brain regions, but they're clinically lumped together. So I think being aware of that, and with that caveat in mind, trying to choose the best models when we're doing preclinical research is, that's got to be step one.
Dr. Robertson, would you like to come in?
Yeah, not easy to follow that. You said a lot there. I'll try to sort of round out and see what I can add to that. But I think a couple of the points are really key, and that is, we're dealing with complex syndromes, and there's not a way to simulate a whole brain, and what happens in those situations. I think that, psychedelics are come from a highly evolved and sort of retained evolutionary system with the serotonergic system. So what's nice about that is even very primitive organisms tend to overlap in the the neural circuitry, the receptology, and allows us to, understand the anatomical relationships and also investigate some of the biomolecular relationships. And also what's nice is some of the same, as you probe those systems or perturb them, if you will, like with pre-pulse inhibition, those types of things also translate well to the neurobehavioral symptoms or outcomes in humans. So it's not a perfect world, but there are, I think we've got 4, 5, or 6 really pretty strong animal models. If we standardize them better so we can compare and design the studies with, a lot of comparative pharmacology as well, using the right comparators, because we know a lot about the system and other drugs, I think you can start to tease apart what's happening in a novel drug and what's also going on with drugs that we, like the psychedelics and like ketamine, for example.
And Dr. Higgins, anything to add to that on preclinical research and drug discovery in general?
Well, I guess my comment here is somewhat shaped by my pharma experience, but a frequent value strength of preclinical research that I've seen includes an ability to conduct PD-PK relationships for drugs, where you combine the measurement of a drug effect in a robust in vivo model or assay and establish drug levels around that assay. I've often seen the translational value that these types of assay designs can bring across multiple clinical conditions. So as an example, in the preclinical space, we've been working quite a lot with drug discrimination, where we've trained rats to discriminate psilocybin from saline using a drug discrimination assay. The psilocybin doses and really more specifically the psilocin plasma levels necessary to generalize. They're equivalent to the plasma levels of psilocin reached in clinical trials at the 25 milligram dose, i.e. around 20 nanograms per ml. The test thus becomes quite a useful approach for looking at novel psychedelics. For various reasons, does an NC generalize to a psilocybin cue?
Mm-hmm.
So does it have a similar receptive state? You can look at things like oral potency of the drug. If you're developing a new drug, you want to establish, if it has oral bioavailability, usually time course of the effect, rapid onset, rapid offset, the time course of a new psychedelic, that's an important thing. And also you can look at plasma levels, of the NC to produce those effects. And often, as I say, I've seen that can be useful to guide the clinical development plan. And also you can set safety, tolerability measures against that. So you can sort of look at the drug properties. So, that's an example of strengths that I can see that, animal preclinical research can bring to sort of drug discovery in this space.
Great, thanks. And not that we want to focus too much on being negative, but to take the other side of that, where do you think that there are weaknesses, or have you seen maybe in your own experience drugs fall down or potential compounds fall down, or how do you avoid kind of pitfalls in preclinical research? And maybe Dr. Robertson would like to.
Oh, punting me the first negative question. I think, there's been a lot of discussion about this, and I think a few things come top to mind. There's the methodology itself and the fact that things are not as standardized as perhaps could be helpful, so that we can have benchmarks and compare across labs, across drugs, across indications. So that, I think that would be one step in the right direction, and that's a weakness. Obviously it's hard to answer this question because I really focus on the strengths and all the great things that do translate. For example, all the examples that just came up. I think that's but the reproducibility, the ability to benchmark and replicate, I think is huge. And then I think it's important when to share the negative data when these models are used, and also maybe not to tweak them so much, to try to optimize and maybe even share the data, the raw data itself with researchers, in the field to kind of help lift all the boats to kind of a higher level. I think the last thing would be just in the design of the studies and the idea that it's good to have converging evidence. So not basing it on just one, but several, and replicating it and making sure you have the right comparators. The big thing about animal studies that I don't know how we can fix is the placebo situation, because we can't really control for the placebo of the experiment. So a lot of our findings are probably overestimated, and that makes it much more difficult to generalize those clinically. And we do see that. We see these really robust findings in the animals, and then it gets less and less strong as we get into the humans.
Dr. Huxter, any follow-ups?
Yeah, this question about the placebo effect is really pertinent, and I mean it's one of those things that comes up again and again, because sometimes your clinical benchmark is your ability to differentiate against placebo. So you could have the lovely effects, but they still might not be better than the sugar pill in a human patient. I don't know if that's a weakness on the preclinical side per se. It's something we need to be aware of. It's an additional route, if you like, to therapeutic benefit. Even you can think of it as a positive, actually, when you're dealing with humans. And certainly there's some discussion that's been ongoing for a few years now about whether or not you just carry on. So you see the placebo effect in some studies, even when the patients know it's a placebo. That's pretty remarkable, right? So, but anyway, aside from that, I mean, I would say, where a lot of the criticism, I think, of preclinical research comes down to the fact that we're working in very homogeneous, very, there's not much variability in terms of the systems that we're testing in compared to the clinical population. And I think that's one of the reasons why things sometimes fall down in stage 1, stage 2 clinical trials. Again, though, whether or not the solution is to introduce greater variability at the preclinical stage, or whether the solution is stratification of your patient population at the clinical stage, that for me is a big question. The other thing that we saw, I mean, not just at Lilly, but anybody who's been working in the Alzheimer's space, has been the issue of when you intervene with your treatment. So in animal or cell culture models, we can throw the drug in any old time. It's not going to be much benefit to patients whose brain is already severely atrophied, for example. This may not be relevant for every indication. I'm just using, here I'm using neurodegeneration as an example. And then the other thing is dose setting. But again, whether that's a weakness or a strength, often when we do work preclinically, we have much more flexibility with the doses that we use. So we may see positive effects at the upper end of the dose scale, or we may set our scales that way. Then when you go to clinical trial, often the tendency is to err on the side of caution. So they'll want to stay well within the safety margins, but you may then be out of the therapeutic margin. So these are all criticisms, problems that I've seen arise over the years. The failure to translate from the preclinical to the clinical scenario. But it's very difficult to pin the blame, on one side of that. It really is, how do we best match them up? It's not so much a weakness per se. It's just, how do we best address those differences? Yeah.
Anything to add, Dr. Higgins?
Well, not very much, I don't think, because I mean, I think it's been, really extensively covered there. I'll just make 2 points. One of assay or model reliability. that can be a weakness. I mean, as I think all of us are aware, you know, some models tests are more reliable than others. And so I think selection of, you know, that can be a weakness for some of the sort of things that we do, placing too much reliance on unreliable models. And the other thing is over-interpreting data that you get from some of these tests. So, you know, for example, interpreting an antidepressant effect of a drug in a very sort of simple test, and you know, placing too much reliance on that, and that can be a weakness. It's not necessarily weakness of the test, but it's just an over-interpretation of the data that you get from that test. So, you know, those are a couple of areas of caution that I would say.
Can I just clarify one thing? With Dr. Huxter, I was thinking of the placebo effect of the experimental methodology, not just the pill. I'm wondering if you have any thoughts about that, and whether you think that also contributes to the, you know, the overinterpretation, perhaps.
On the clinical side, or the
On the animal side, because we don't. That's one thing we, you know, there are site differences, and we can't control for the lab differences and those others. So is that also something where you see these great results? Something that the lab is doing?
I mean, we, as a company, we operate across multiple sites, and we're acutely aware of the fact that, you know, sometimes it can be a challenge replicating results in somebody else's hands, and you may never know exactly what it is. It just takes a lot of hard work to establish that replicatability, you know, and not just within an organization, but then especially, you know, if you're dealing with multiple companies, if you're trying to put together a package of preclinical evidence to support your program. Yeah, I guess it's just the standard things we try to do, and that's, you know, to follow, you know, very, very well-defined protocols, blinding wherever possible, of course, to eliminate, you know, experimenter bias. But the question about, you know, what is it that, I guess you're less concerned about whether a tissue culture is going to respond to whether it's a male or a female person in the experiment. You never know. But certainly with animal experiments, if you change operator, you change the person who's running the study halfway through, you can certainly see, you know, effects there. That's just the general pantheon of potential confounds to your experiment that we have to deal with every day. You know, somebody goes down the corridor with a loud trolley, you know, in the middle of a sensitive experiment, throws everything off kilter. Yeah, we've all been there.
Yeah, so actually just following up on that, I guess that the next question I was kind of going to ask was around, you know, a lot of these, particularly with psychedelics, you're kind of looking at modeling neuropsychiatric conditions, which, as you've mentioned, you know, is quite difficult, or, you know, in the general sense, some might say maybe not impossible to do. But how best do you go about modeling these conditions in animals? What are, I mean, you have touched on a few things already, kind of avoiding pitfalls, but what kind of methods would you think or recommend how you do that best? And maybe Dr. Higgins, if you want to jump in.
Well, I certainly have the view that the best approach for trying to model complex neuropsychiatric conditions is to try and break them down into specific sort of diagnostic criteria and develop animal tests or models around that. So a good example would be attention deficit hyperactivity disorder, where you've got two, or you know, at least two core criteria of inattention and poor impulse control. And so you can tackle those aspects separately by specific animal tests and look at you know, animal behavior in that. You can impart challenges to those tests, to tax attention or impulse control and so forth. And I think that's going to be a far more fruitful way of actually investigating drugs, you know, to treat these conditions. And what you might well find, you know, is a drug that produces, say, good impulse control, not necessarily a big improvement in attention, but that drug could then also have potential across, you know, other clinical conditions. So, you know, and also, you know, a lot of these tests are translational, so they have, you know, human equivalent tests, and I think, you know, that provides a further value in that, you know, I think you've got far more chance of a drug effect translating from what you see in an animal into a human if you're doing basically essentially the same test, you know. So you know, that I think, and it's obviously increasingly being adopted. I think that's very much believe that's the way of the future. You know, focusing on endophenotypes of these complex clinical conditions and aligning preclinical models to the human.
And I think the proof of that is, you know, you see it in the sheer volume of off-label, you know, prescription use of existing drugs across multiple indications. They weren't necessarily developed for, but you know, you see it all the time, especially in neuropsychiatric disorders. You often get antidepressants and antipsychotic medications being prescribed for each other, vice versa. You know, because they are complex. They are complex conditions.
Yes, and they're being used to treat a specific aspect, you know, of that condition as well, typically. Yeah.
I think that linking the molecular neurocircuitry and the molecular research, you know, at the receptor level with all of the off-target activity and on-target activity to these specific final common pathways, if you will, and then to the indication, I think is a nice approach. Because, you know, we're really seeing that you can tweak the chemistry just a little bit and get a whole different profile, you know, with the heterodimers and all of these different modulation pathways. So I think that's going to be another key is even the nosology. You know, we say SSRIs. Well, we know so much more now than we did a decade ago. So being able to take across species data, across modalities from the micro to the macro, I think by the time you get to the clinic, if you've done that, you may have a pretty good idea of what symptoms you might target, and then that composite of symptoms probably makes up a disorder, right? But the one receptor system that throws it completely off for use in a certain indication. You know, if you happen to cause some anxiety, that's probably not going to work for a lot of things, or you don't hit impulsivity, for example, in ADHD, but you hit just attention. So I think those are the other key elements, is to begin with the end in mind, like you're saying, and think of what you're targeting, and then test that in your models, our models.
Yeah, that's great. Some really great insights. So to move on, I suppose, now to actually the psychedelics in this space. So these compounds they're already kind of in the clinic, and they've been around for a long time. So, you know, kind of mechanism of action is known, and structure is known, and effects, and they're not kind of taking that classic maybe drug development route. So does that change how we address them in a preclinical sense? Do we test them differently than something that's coming through for the first time, or is the approach different at all? Maybe Dr. Huxter?
In terms of whether or not we're like treating an analog of an existing substance which is sort of used recreationally or otherwise, or versus a truly novel chemical entity, do you mean in that regard?
Yeah, I mean, either or. I'm just kind of spitballing, I suppose, ideas. I mean, is it just a case, do you just supplement, say, like, you know, a psychedelic with something that's already being tested and see if it works? You just shoehorn it into already established things, or do you address it slightly differently because you have all this added information?
Well, I think I suppose you start from the assumption that you're targeting the same mechanism, right? So with any of these drugs, you pick one. I mean, it might be dissociative, it might be a psychedelic, it might be, something else, but you start probably with a hypothesis, a receptor or a pathway that you're going for. It could be 5-HT2A in the case of psilocybin. And presumably if you're hitting that, but it does, depending on how different the chemical entity is, would depend on what kind of background, what kind of story you need to build before you really start running intensive, say, an animal study, because you really do have to understand whether it's going to affect the PK of the compound, the time at which you see your maximal effect, what your half-life is. If you were to embark on an animal, a big expensive animal study without knowing those things in advance, you could be completely missing your therapeutic window, both in terms of time and dose. So, I think the further you are away from your original, your compound that you have well characterized, probably the more homework you need to do initially to establish, what is the blood-brain penetrance, what is the receptor occupancy, is it metabolized differently? And then once you know those things, you're not going to cause harm if you're doing it in an animal study, and B, you've got some idea when you should be looking to see your effects. I think that's the main difference when you're going in. I mean, from a clinical perspective as well, the more different you are from some of these drugs, which have been like ayahuasca or psilocybin, which have been recreationally used we kind of know what the safety profile is. Whereas if you're really tinkering a lot with the molecular structure, you may have to kind of make sure that you've established, liver toxicity, or I don't know, maybe that doesn't really apply for this class of drugs, but you know what I mean. You need to make sure that it's not going to fall down at the clinical stage because some horrific tox emerges. I mean, that's why probably 80% of drugs never make it. They fall down at the tox. So that's something we need to be aware of if we're changing things too much.
Dr. Higgins, if you'd like to go next.
Okay, so I guess we're coming at repurposing established depression assays, or how we look at psychedelics in depression tests. There's something we haven't touched on is in terms of psychedelics, there's both a macrodose and a microdose idea. Obviously, the macrodose is far more developed, far more established and accepted, but the microdose, I think, is a really interesting phenomena as well. But taking the macrodose from the animal modeling perspective, psychedelics are given in a very unique way. They're given on 1 or 2 occasions in combination with behavioral therapy, and then months, potentially longer afterwards, you've got a change, a behavioral change, a clinical effect. So there's a real disconnect between, what I was talking about earlier with the PK/PD relationship of the drug. And of course, most of us have been sort of working up to now just on looking at drug effects, in animal tests with the drug on board, not specifically looking at enduring changes. So of course that, that's a very big difference, and I think that needs a bit of a sort of rethink, and obviously, that I think some of the tests can be redesigned to accommodate, just looking at longer term changes on behavior. Sorry, looking at longer term changes on behavior. The microdose is quite different, and we've done some really interesting work on this. And microdosing is sort of, it's the psychedelic is taken at a low dose, about 1/10 of the psychedelic dose. It's taken on a more regular basis, and there isn't a psychedelic state associated with it. And And so there could well be a sort of closer correspondence between therapeutic effect and the actual, drug exposure. And we've done, we've seen some really interesting effects of low dose psilocybin as well as other psychedelics such as DOI on, things like motivated behavior and attention in poor performing animals. And I think, that some of these effects seen at low doses may correspond to what might be picked up in the clinic under this sort of microdose umbrella. Of course, the big problem at the moment is the lack of clear, placebo-controlled trials to really sort of nail this phenomena down as a clear clinical property. But it's interesting, and as I say, in behavioral models we can they're subtle though, and so it requires high end, high study ends, and maybe picking out performance subgroups, poor performers, and that might have some equivalence to the clinical population who might benefit, from the low dose therapy. So, I think, to address that question, I think the idea of micro and macro dose, is a different answer to both, but it provides an interesting question.
Dr. Robinson, do you have anything to add?
I think that the psychedelics offer them kind of a wonderful wealth of information relative to developing a novel compound in this space. Because as we kind of mentioned earlier, the slightest tweak in the chemistry can send you down a whole different path. So having all of this data, a lot of it is anecdotal. I appreciate that. Really gives you a head start, I think, on knowing about the safety and the dosing and some of those relationships. But to get to the clinic in a formal way for drug approval, I mean, there's a lot of backfill that has to be done. You still have to do all the same work that you would have had to do from the beginning if there wasn't all of that data. But you might be able to do in parallel and accelerate the development. I think the point Dr. Higgins brought up about the durability of effect and the difference between high and low doses, because we know that also perturbs the system differently, and we might get a whole different response at a higher dose versus a lower dose. I think the other part about the durability is understanding what's going on in the brain, and not just histopath, like, are we having cerebellar atrophy or something like that, but for the placebo effect that you mentioned, or even for side effects. So I've thought a lot, hey, we need to combine imaging and same as the animals, combine these modalities. So, if a subject says they're having or they report a certain adverse event, and it's a phenomenon we can't really see, if we're also imaging those same subjects, either with EEG or, imaging, maybe both, we can also look at what's going on in the brain, and that will also kind of help us kind of reconcile those disparate events and make sure that we're reporting those things properly, and then making our scales more sensitive, I think, as well. I know a couple of the companies are working on that, and I commend them, because again, even our nosology, I get asked all the time, is this a hallucination or is that a hallucination? What's the difference between a hallucination and a visual effect? Is the hallucination the trip? And I think those are, maybe for some folks it's splitting hairs, but I think those semantics are going to become fairly important, because we're going to need to measure them to differentiate.
And actually,
way more than you, way extended before you went, but a lot of grist for the mill here.
I was just going to say, do we want to talk a little bit about what we would consider a hallucinogenic experience in a preclinical model? I mean, that's it's a little bit tricky. We have some, obviously, that we routinely use. I'm not going to, I'll leave it to, oh, okay, I'll go ahead and say it, head twitch. So head twitch is one of the standard assays that we use. Are we, suddenly I'm the moderator, feel. Are we happy that twitch is actually doing what we think it does? I mean, I think the receptor occupancy data is there to suggest that you see these effects, but again, we know that the dose relationship is complicated. It's not linear. Whether or not that falls into the microdosing range to your question, Guy, I'm not sure. We tend to see this inverted U-shaped response, perhaps at things that would be beyond what we would consider a microdose. So it could be genuine off-target effects. But with regards to how we determine if an animal, for example, is hallucinating, what do we all think of the current, I would say probably gold standard, which is the head twitch in mice or wet dog shake in a rat?
I can, yeah. Just to address an earlier thing you said about how does a low dose compare with, those cardinal 2A signs of wet dog shakes or back muscle contractions in rats, head twitches in mice. We basically initially focused our studies on the very low end of the ascending dose response curve of those effects. So, taking your, comment that perhaps the head twitch is a wet dog shake as a proxy for the sort of hallucinogenic effect. I'm a bit of an operant guy, I guess, and I like operant tests, and for their reliability and so forth. And I think drug discrimination is perhaps the one that has the best face validity in my eyes, in terms of what you're doing. You're training a rat to a known hallucinogen, be it psilocybin or LSD. And then you're asking the rat, well, does it see drug X as similar? So you're getting at the internal state that the drug is generating. Now, whether that's a hallucination who knows? They won't tell us if they're seeing tangerine trees or marmalade skies. They won't tell us that. But what they are saying is that they recognize the test drug to what they've been trained to, and you see cross generalization between things like LSD and psilocybin. We've generated. What's interesting is amongst the different psychedelics. So we're also beginning to explore, and other groups, GATCH for example, looking at other discriminative cues engendered by, say, DMT or 5-methoxy-DMT and others, looking at cross-generalization. And there does seem to be some differences, which may well reflect the different pharmacologies of these drugs. So, for example, 5-methoxy-DMT has got more 5-HT1A agonist property, which may contribute to its overall cue and change subtly the state. So what we've seen is partial generalization between some of these drugs. So I think my personal view is drug discrimination is the best that we've got at the moment to look at a psychedelic state in preclinical species.
See, I think also we're starting to see some interesting consistency in what we pick up with EEG as well when these drugs are on board. And for me, throughout my whole career, has always been a bit of trying to bridge that gap between human imaging studies and the kind of EEG or local field or even single cell recordings that we can do in animal models. And it's never easy, right? Because it depends on where your electrodes are. It depends on what your sampling rates are. It depends on a lot of things. But one of the things that gives me some encouragement with regard specifically to the psychedelic experience is that, David Nutt and Carhar Harris have published a couple of papers now with fMRI looking at functional connectivity and bold imaging. And, the emerging story is that you see this sort of transient suppression of brain activity and again, connectivity, especially in the lower frequency bands. And lower, I mean, sort of below gamma frequency range. And this is something that we see again and again, and it follows the same sort of time course as you would expect with, maybe they had twitch responses a little bit faster, but certainly these are acute effects of having these drugs on board in rodent EEG. And, we've seen it enough times now that I'm starting to feel confident that I can look at a spectral power analysis and say, that's an animal on psilocybin. Whether or not he's hallucinating is another thing, but certainly the time course, the dose, and from the human imaging study, what people report when they're having a psychedelic experience and how that maps onto, these kinds of effects in both EEG and connectivity. To me, I don't think it's a rock solid story. It's still emerging. It's still fairly new, but I think the story is emerging, and the more data comes out, the more confidence we'll have with these other physiological measures, which maybe allow us to tell whether or not, the animal is experiencing something equivalent to hallucinations.
Yeah, I think these drug signatures, as you're referring to, I think are going to be really helpful for second, third, and fourth generation drugs, right? And being able to recognize a psilocybin signature profile for any of these different assays, I think is going to be a huge step forward.
Yeah.
It's nice to see that the conversation is going this direction. The next question I was going to ask was related to the hallucinogenic kind of experience, and can it be modeled, and then also whether is it necessary to model it. I know obviously Delics have a specific kind of goal in mind and some people, I think it's quite an interesting debate at the moment whether how necessary is the hallucinogenic experience to it all. So I don't know, Dr. Robertson, if you'd like to speak to that at all, of whether is this hallucinogenic experience necessary?
So I think what's really important is that these are my opinions, and I hope they're completely in line with my colleagues at Delics and amongst the field. But I think at Delics, we don't really believe there's a one size fits all. And as a physician scientist, I especially think that different patients may not only need different types of treatments, they desire different types of treatments, and many would, whether they could benefit from the experience or not, they may not want that experience, and they may have other risks that prevent that. So when I think about at a mechanistic level, is it necessary? I think it's already been proven that it's not necessary for an antidepressant effect, because we have SSRIs and we have other drugs. As we understand more how the psychedelics differentiate, I mean, we're just kind of starting, right? We have some treatment-resistant depression studies. We're getting some anxiety and PTSD coming out. Very small. The anecdotal evidence, I think, was like, I don't know, we were all waiting with bated breath. Is that going to happen and generalize? And a lot of it is generalizing, but not all of it. So some of our effect sizes seem to come down. What seems to hold true is this onset of action, and possibly also the suicidality component in response. I think based on other examples, we know that you don't have to have an experience to also benefit in that direction. Where that makes a difference, I think that's yet to be seen. I think it does make a difference to many people, and I think in the neuroanatomical sense, I sort of think of PTSD and what some of the patients were saying after they did a nerve block in their neck. It was the first time they could actually just relax and kind of disconnect from the stress of the world and all of their thoughts and everything. How that happens, whether it's peripheral, central, with or without hallucinogenic effects, I think it's core to the experience of healing. And I think we've got some work to do to see, you know, which patients will do the best with which types of experiences and what key components of the experience are most critical to what patients. I don't know if that's a big, these are big questions.
It's tough to fit all this in an hour.
Yeah, you know, I think we're doing a lot of good work here today, but we'll probably have a lot to think about after this.
Yeah. Yeah. Any other comments on how necessary the hallucinogenic experience might be?
I think this reflects, you know, part of the fun of what we do, right? That I would say, you know, obviously a lot of people believe that the psychedelic experience is tied to the therapeutic effect, but maybe not, and we will know you know, once the clinical trials are done. I mean, you know, a big part of the clinical effects seems to be this disruption of, you know, ruminative thoughts and then the learning of new behaviors. Is the psychedelic experience necessary for that, or just, you know, having a capability of having some effect on brain neuroplasticity to allow new behavior, you know, is that sufficient? You know, this is the fun, I think, of in terms of what we do, and you know, when you're in a developing area of neuroscience and you're trying to sort of think of animal models, you know, to model these complex clinical conditions. Ultimately, you've got to get to the clinic and get there as quickly as possible, and that will tell us, you know, hopefully by well-designed clinical trials. The jury's out at this moment, right?
And that was actually going to be, oh, sorry, Dr. Huxter, go on ahead.
Oh, no. I mean, I was just going to say it just kind of highlights how, you know, our state of understanding the mechanism of action for these drugs is still very early on, right? It really highlights everything that we've discussed here, really highlights that that's why it's such an exciting area scientifically. Mm-hmm. We really have so much to learn about how these drugs are actually doing what they do and how we can differentiate those different effects. That's all. I was just going to say that.
Yeah, just one last kind of quick question I was going to pose to you. And from a preclinical point of view, I mean, obviously there's been a lot of focus on psychedelics with depression, but where do you see kind of other kind of conditions coming in, and how difficult will it be to kind of see the effect of psychedelics in those? I'm not sure if you have any experience yourselves with different outside of depression kind of assays and things like that.
There's a couple of clinical trials ongoing around anorexia, for example. PTSD, Dr. Robertson mentioned there. And I think, Guy, you sort of probably touched on it, actually, when you said things that involve ruminative types of thoughts and behaviors seem to be, you know, amenable to this mode of therapy. I've also been in interesting discussions about whether or not there are certain types of chronic pain which might be with these kind of therapies. Once it becomes centralized, once, you know, there's no obvious trigger, it has more to do with, you know, how we interpret and process external signals coming in. So I mean, those are the ones that come to mind, but yeah, I mean, it could be way beyond treatment-resistant depression, I think.
Yeah, the one I would add to that is sort of various addictive disorders. So, you know, such as, you know, people seeking treatments for smoking cessation. There's some interesting data, you know, albeit in relatively few individuals, but, you know, smoking cessation it's been, even Chantix is only sort of about 30%, I think, of people are abstinent after about a year, you know, so it's got a very sort of low efficacy bar. But some of the early data with psilocybin from Roland Griffith's lab is showing, abstinence rates significantly higher. They were almost double, I think, sort of up in the 60% range. Now, obviously, that needs to be, studied in greater numbers and so forth. But, that's an area where you can see some, perhaps some very, significant therapeutic potential of psychedelics, as well as the others that John said, and some really interesting ones as well.
I was thinking that there's a,
Pain.
okay, I mean, I'm crazy, right? I mean, look at all of the different things that clinicians are using ketamine for now. Migraine, sort of fibromyalgia, the neuropathy, to your point, chronic pain. If you're hitting the receptology, depending on your compound, may overlap in the antipsychotic space, the antidepressant space, the stimulant space, anxiolytic space, and depending on how that Venn diagram comes together, I don't think we've even tapped the potential yet of where these drugs might be helpful.
Yeah, I mean, there's a real unmet need as well, in these areas. I mean, eating disorders, anorexia, it's the highest mortality rate of all psychiatric indications. I mean, it's a massive problem, and there are no drugs actually that work, and most of the ones that are in use are off-label. They're not developed for it. They're trying to tackle aspects of the of the symptoms. So yeah, it will be very interesting to see what the results from these. There's 2, I think, clinical trials ongoing.
Yeah, John, you bring up such a good point. Is the young people, because suicide's a 3rd leading cause of death now in people under 24. So, when I think of TRD, but when I think of suicidal ideation and suicide attempts, that's where I think we get anymore. There's a pretty strong effect there, but we'll have to talk about when do we start looking at the more developmental trajectory.
Yeah, I
Great.
agree.
I think I'll jump on. I'll try to get a couple of questions answered maybe before we finish off. So just scrolling through here, this interesting one here. When considering hallucinations in animal models, should we consider the sensorial dominance or anything in particular to the animals? So things like smell or auditory versus visual hallucinations. Would that be a factor? I don't know who wants to try and answer that.
From the physiologist's point of view, you measure what you're able to measure, right? So I think we see what we see because of our particular placement above the frontal and the visual association cortices with our electrodes. So to me, it suggests something that may be visually related, but we're simply not measuring from the auditory cortex, for example. So I have no way of knowing. I'm not sure what a head twitch response might tell you about whether or not there's sort of modality to the experience. Do you have any thoughts?
Not really. No, I kind of treat that as a pretty characteristic motor response that clearly reflects target engagement, but actually interpreting the mechanism. There was some work with the wet dog shake behavior from Herb Meltzer's lab, I think, looking at microinjection of DOI as a 5-HT2A agonist into the prefrontal cortex and reporting a head twitch response. But in terms of, other mechanistic studies where they've sort of tried to look at, what the neuronal structures that underlie that behavior, that's about the only microinjection study I can think of. And it does suggest there's a prefrontal cortical locus for that effect, which of course is pretty densely packed with the 5-HT2A receptors.
Is there a difference between the shake and the twitch and the shake? One thought to be a little more dissociative, one is one is more maybe hallucinatory, visual. But I think that for me, the challenge with when they bring up the olfactory system is it's just so different in animals. That those sensory distributions in the brain are quite different than humans, so that's a little bit harder to translate to your point. But I think the visual cortex, a lot of that is reserved, so you can track those types of phenomena much easier. I think I'm trying to think of a study too that you were talking about where they were able to somewhat connect to the rodent's visual cortex and get an idea of what they might be seeing. And it seemed like it was a distortion of shapes is what kind of came out of that, I believe was psilocybin.
That's interesting.
Yeah, I'm pretty conservative with some of these things. So, you know, I do tend to interpret, you know, these motor effects as, you know, just you know, a signal of target engagement. I think there's definitely value in the head twitch response. It's centrally mediated. It's if you I mean, other drug classes can induce head twitches, so I think it's important to do some sort of pharmacology on that. And, you know, obviously there are very good selective serotonin 2A antagonists, such as M100 907. And if you show that drug at a relevant dose blocks the head twitch, it's a very good sign that your drug of interest is hitting 5-HT2A receptors. In terms of what it really means in terms of, you know, the state of the animal, I don't really go beyond that. I must admit, I'm pretty conservative in how I look at that behavior. Another thing is it's very disconnected, the head twitch response, from the drug exposure, because you were talking about that bell-shaped dose response curve, John, and you know, the decline is, I'm damn sure, is because other behaviors are kicking in which actually oppose the expression of the head twitch response.
It just kind of freeze at some point and stop moving, right? And so,
yeah, they do. That's exactly right. They become immobile. Yeah. Francie Grossi did some nice stuff showing that if you block 5-HT2C receptors, you can expand that dose response. So, you know, I think that's these drugs aren't particularly selective, right? And so psilocybin, you know, it hits. It's a natural drug. It wasn't designed as a 2A agonist. So,
you know, I was thinking what you were talking about in the training of the animals, and I believe in that study I mentioned, the way they were able to understand what the animals were seeing was they trained them to the shapes, and then they gave the drug, and they measured those behaviors, and they were quite different. And that sort of said that they could make out certain things, but not others, and you know, other parts of their sensory system remained intact. So that might, I think you're onto something with the training and then adding the intervention. I think that makes a lot of sense as far as a preclinical design to kind of probe something specific like that. But yeah, I agree with you. The locomotor definitely could impact what override what you think you're seeing cognitively, right?
I guess we could use things like, you know, auditory mismatch negativity and steady state response to look at auditory processing as well, to see if there's disruption in that modality.
Yeah, no,
another quick question here for Dr. Higgins, just to bring it back to the microdosing. Are you able to establish a discriminative stimulus on a low dose of psilocybin, and does it differ from the macro dose? So if you try doing a microdosing,
that's a really interesting question. That's a really interesting question. So we published a Frontiers paper on some of this work, and we found that the sort of high microdose, if that makes sense, was around 0.1 milligrams per kilogram of psilocybin, and that corresponds to plasma levels of sort of 7 to 12 nanograms of psilocin. Now we've trained rats. We haven't trained rats at doses lower than 0.5 milligrams per kilogram. And so the 0.1 mg per kg dose is very much at the bottom end of that dose response, of the generalization dose response curve. So you might get maybe 10% generalization. I suspect that, I mean, most published work with psilocybin uses slightly higher doses of 1, and I mean, you know, there's you know, 1 milligram per kilogram. I think if we go much lower, we're going to struggle with reliable stimulus control. So although we haven't tried training rats to a microdose, and I think the question is a really good one, I suspect that we will not get reliable stimulus control at the psilocybin dose, you know, 0.1 milligram. I just don't think we would get it, at least not in a significant proportion of animals, maybe, you know, 1 or 2, but not enough to do, you know, that we had to go to heroic numbers as well. You know, we did this low responder for progressive ratio. We had 72 rats trained, and we took the lowest tertile, so that was 24 animals, and there we could pull out and, you know, a significant effect increase on breakpoint and in an attentional task as well. The effects of, sorry, Dr.
No, go ahead. You're just breaking up a little bit.
Oh, am I? Oh, sorry. Okay, well, I basically, you know, to answer that question, I don't think we could get reliable stimulus control training rats to a low psilocybin dose in what we kind of call microdose range.
Just checking the time there, we're just about at 3 o'clock, just gone over slightly, although I feel like we haven't solved everything, but it was a very enjoyable chat. So I would just like to thank all the panelists for their very valuable time, and thanks everybody who tuned in to watch. So hopefully we'll see you all again next week at the last webinar. Thanks for your time.
Thank you as well. It's nice interacting with you all. Bye-bye.
Yeah, me too. Thank you. Bye-bye.
Nice meeting you.